All right, welcome back. This is the first lecture of the final series for this course, and we're going to talk about early brain development. So early embryogenesis involves a process called gastrulation. Gastrulation is the establishment of the basic body axes. So we've talked about that before. One moment. There, I'll just pull those up. We've talked about superior and posterior, anterior, inferior, dorsal, lateral, dorsal, ventral, and lateral is away from the midline, anterior or rostral, and posterior or caudal. So this is the diagram that's going to guide the next several slides as we talk about gastrulation and the establishment of the basic body axes and early embryogenesis in general. So at this stage, there's a development of 3 layers, so 3 embryonic germ layers from the blastocytes. And these are called the ectoderm, the mesoderm, and the endoderm. And they're named for their relative position, which I'll get to on the next slide. But each layer gives rise to specific sets of tissues or organs in the body. So each of these layers is the first stage for everything else in the body. So here are our germ cell layers. The ectoderm is the outer layer of tissue, and this creates or grows into the nervous system and the skin. So ecto means outer from the Greek, so the outside. We have the ectoderm. The mesoderm is the middle layer, so meso meaning middle. This gives rise to blood vessels and kidneys. And it initiates the invagination or the folding in, so being turned inside out or folded back on itself to form a cavity or a pouch. So the invagulation that defines gastrulation. And we'll get to all of this as well. The endoderm is the inner layer of tissue, so endo from inside. Endocannabinoids, for instance, are cannabinoids that your body naturally produces, and exogenous cannabinoids are like outside, like THC, for instance. So the endoderm, so the inner layer of tissue, creates the lining of the lungs and, well, grows into the lining of the lungs and the liver. So here is our orientation, and this is the growth that we're going to be talking about over the next few slides. So dorsal up here, ventral down there. This is anterior and posterior. The notochord right there, this is formed at the midline of the gastrulating embryo. It's a cylinder of mesodermal cells. So it's cells from the mesoderm that's generated at the site of the surface indentation, so known as the primitive pit, that eventually elongates into the primitive streak. And the notochord defines embryonic midline. So it defines the midline of the embryo, and it's the axis of symmetry for the whole body. It's a transient structure, which means that it doesn't last. It disappears following early developments, and it sends inductive signals to the overlaying ectoderm. So there are signals from the notochord that trigger activity in the ectoderm. And the ectoderm that's immediately dorsal to the notochord is known as the neuroectoderm. This gives rise to the entire nervous system. So the ectoderm layer that's immediately dorsal to the mesoderm cells that form the notochord is what eventually develops into the nervous system. So now we're at about 18 days of gestation, and that's when the process of neurulation is underway. So that's when those inductive signals from the notochord of the mesoderm trigger the neuroectoderm so that ectoderm region immediately dorsal to the notochord, to differentiate into a subset of cells called neuroectodermal precursor cells. And these cells here, they thicken and they become what's known as the neural plate. So as the neurulation proceeds, the neural plate begins to fold at the midline. So again, adjacent to the notochord, and this forms the neural groove and ultimately the neural tube. So the neural plate that's immediately dorsal to the notochord here, that differentiates into what's called the floor plate, and the neural crest is the what emerges at the lateral margins of the neural plate, so furthest from the notochord. So you've got the neural crest, the lateral margins of the neural plates, and these come to get closer together as the neural groove develops and then eventually forms the neural tube. Now, it's important to know that the neural tube, it's not homogeneous. There are different cell types within the neural tube. For instance, the ventral portion here, the floor plate, this is a specialized strip of epithelial-like cells that provide the signals for neuroectodermal precursors of the spinal cord and the hindbrain. And the neural crest emerges at the lateral margins of the neural plate. So once the edges of the neural plate meet at the midline, the neural tube is then complete. So what was the neural crest becomes sensory and autonomic ganglia, so it becomes the peripheral nervous system, so here. And then the mesoderm that was adjacent to the tube, it starts to thicken and it subdivides into somites. So these are precursors of skeletal and muscle tissue. And the neural tube adjacent to the somites becomes the rudimentary spinal cord. And then the fluid-filled cavity of the neural tube, so this is full of fluid, that becomes the ventricles, the ventricular system within the brain. And then finally, the anterior end. So again, this is anterior. You can look up here at our little axis. The anterior ends of the neural plates, so known as the anterior neural folds, these grow together at the midline, and it continues to expand and eventually gives rise to the brain. So here's the neural groove and the neural tube. These crest cells are now not part of the neural tube, but the stem cells of the neural tube ultimately give rise to the entire brain and spinal cord and much of the peripheral nervous system. Now, the floor plate and the roof plate, these are, again, transient structures, but they signal stem cell differentiation of the spinal cord and hindbrain development from the neural tube. These neural crest structures, they give rise to the neurons and the glia of the sensory and autonomic ganglia. And they do this via 4 distinct migratory pathways. So these neural crest cells can differentiate into many different types of cells. The ones that migrate along pathway 1 here become sensory ganglia, and the ones that migrate along pathway 2 become autonomic ganglia. Those that migrate along pathway 3 become adrenal neurosecretory precursor cells and end up aggregating around the dorsal kidney. And the neural crest cells that migrate along pathway 4 become non-neuronal pigment, cartilage, bone, primarily of the face and skull. So these are non-neuronal cells. Now, soon after the formation of the neural tube, major brain regions gradually begin to form. So they begin to differentiate and change in shape and cause bends and folds, and there's constrictions of the tube. So At the anterior end here, there's a crook. So this is known as the cephalic flexure. So it's a crook. And the cephalic flexure balloons into the prosencephalon. So this is what gives rise to the forebrain. So think frontal cortex. The bulge that forms rostral to the cephalic flexure, so here, and posterior to the prosencephalon is the mesencephalon. So this is a cross section of this diagram here. So the mesencephalon forms here. So it's rostral to the cephalic flexure and posterior to the prosencephalon. So it's this region right there. Now the rhombencephalon forms between the cephalic flexure and the caudal cervical flexure. So this region here in red, so cross section to there, that gives rise to the hindbrain. And the neural tube that's caudal to the cervical flexure is what forms the spinal cord. So focusing on the telencephalon and the diencephalon. So the telencephalon is the lateral aspects of the prosencephalon, and this grows into the cerebral cortex and the hippocampus and the basal ganglia, the basal forebrain, and the olfactory bulb. So now look at our cross section and how much more complex that's starting to look. Then the diencephalon here, which is the caudal portion of the prosencephalon. So now the prosencephalon has grown into two distinct substructures. So this diencephalon eventually forms the thalamus, the hypothalamus, and the optic vesicles, including the neural retina. And now focusing on the mesencephalon, the metencephalon, and the myencephalon. The mesencephalon retains the same name from early gestation, and this structure eventually grows into the inferior and superior colliculi and the midbrain tegmentum, including the substantia nigra and ventral tegmental area, which we've talked about previously. The metencephalon here, this is the rostral rhombencephalon, and this grows into the cerebellum and the pons. And then the myencephalon, this is the caudal rhombencephalon, and this grows into the medulla. Now, the neural tube is organized into repeating units called neuromeres. And this leads to the segmentation that establishes regional identity in the body and possibly also the brain. So there's parallels between Drosophila segmental genes, so the inferred ancestral homeobox genes from which invertebrate and vertebrate segmental genes evolved, and human Hox genes. Now, Hox genes in the hindbrain and spinal cord, and DLX and PAX genes in the midbrain and forebrain, these are responsible for guiding differentiation of the embryo into distinct segments. So Hox genes of humans and of most mammals have apparently been duplicated twice, leading to 4 independent groups, each on a distinct chromosome. And the anterior to posterior pattern of the Hox gene expression is reflected both in flies, in the Drosophila, and in humans. And this pattern is what regulates the morphological features. So all of these bends and shapes and folds that we express that underlie the progressive regionalization of the developing neural tube. So just be aware of this notion of segmentation of the neural tube. The embryonic notochord and floor plates and roof plates and the adjacent tissues like the somites, these produce molecular signals. that induce cell and tissue differentiation in the vertebrate embryo. So this is known as neural induction. These are the instructions that establish stem cell capacity to make nerve cells specific to each region. And these produce molecular signals that induce cell and tissue differentiation in the embryo. So in experiments conducted 100 years ago, they cut out sections of developing embryo and transplanted the tissue to a different region. So cut out a bit of the notochord and transferred it to the roof plate. And the cells that were transplanted, those either acquired a new identity and were influenced by the local factors, or they retained their original identity. So they were influenced by original or immutable instructions. So there can be interactions between cells that influence the cell fate. So these are local diffusible factors and also direct physical interactions between membrane proteins. And the embryonic notochord, floor plate, neural ectoderm, somites, these all produce molecular signals that induce cell and tissue differentiation. And the results of these inductive signalings include changes in gene expression and shape and motility of target cells. So there is a lot going on in the embryonic neural tube that is necessary for proper formation of our central nervous system. So here we have a cross section of the neural tube, and we have the ventricular or the ventricle. So this is the inside of the neural tube, and this is the outside. So this is known as the pial surface. And then this is known as the ventricular zone. So if we take a section of that and put it here, so this is the innermost section, the ventricular zone, and then this is the outermost section, so the pial surface. So neurogenesis begins after the initial patterning of the neural tube is complete. So once the neural tube is where it is and cells have started moving and the patterns all there, then precursor cells can start to develop into new specific types of cells. So precursor cells have distinct signatures of gene expression that provide basic identities. These precursor cells are located in the ventricular zone, so this innermost cell layer that surrounds that ventricle, the inside of the neural tube. And these cells undergo a specific or stereotyped pattern of movements, which we'll get to in the next slide. And these movements lead to the formation of either new stem cells or neuroblasts. So neuroblasts being immature nerve cells. So stage 1, G1, this is the growth stage. So this is when the nucleus of the cell is near the ventricular surface. So to orient you here, this is the inside of the neural tube, so the lumen or the ventricle, and this is that outer pial surface. So at growth stage 1, the nucleus is near the ventricular surface here. And then at stage S, this is the DNA synthesis stage, so S, the nucleus is migrating toward the pial surface, and the DNA is replicating at that point. So now we get to growth stage 2. The cell is growing, the nucleus is migrating back toward the ventricular surface, and the precursor cell loses connection to the outer surface. So it loses that connection here. And then finally, there's mitosis. So there's two possible outcomes of mitosis in this case. The first is symmetrical, and that's when it produces two neural stem cells. And the second is asymmetrical. So this is when it produces another neural stem cell that re-enters the growth cycle, along with a mitotic neuroblast. So asymmetrical mitosis results in a new stem cell and a neuroblast, so an immature neuron. And symmetrical mitosis creates two more neural stem cells that each then undergo this growth, synthesis, growth, and then mitosis. And at its peak, neurogenesis, the human brain is forming 250,000 new neurons a day. Yeah, and so I definitely made a mistake on the last slide. It's not 250,000 new neurons a day. It's 250,000 new neurons per minute during peak cell proliferation. Anyway, so this set of images here is time-lapse microscopy. And this allows us to see the division of a precursor cell into two cells. So whether it's symmetrical or asymmetrical, I'm not sure. These arrows point to the radial processes of the cells, but it's out of focus of this image. So now we have these neuroblasts that have been formed through mitosis, and these are progenitor cells that can become either neurons or oligodendrocytes or astrocytes, but they need to migrate. And some of these, well, most of what we talked about so far has all occurred within the ventricular zone, but these cells need to migrate outside of the ventricular zone. And most of them, well, some of them will use existing axon pathways to guide migration, but most that have to migrate long distances use radial glial cells to guide them. So there are these glial cells that the neuroblasts will use to migrate to where it is they're supposed to go. So these post-mitotic neurons adhere to glial processes, and you can see here again with microscopy the movement of the cell, migration of the cell outside of the ventricular zone. And they use these processes to move beyond the dividing cells in the ventricular and subventricular zones and past already differentiated neurons in lower cortical areas of brain structures that express layered or laminar structures like the cortex or the hippocampus or the cerebellum, etc. Now we have a video.
Shortly after fertilization, the fertilized egg or zygote begins dividing. By about 4 days after fertilization, a solid ball of cells called a morula forms. The cells continue to multiply, and by day 5, the cells separate into two groups, the inner cell mass, which will become the embryo, and an encompassing sac called the trophoblast, which will develop into the placenta. Trophoblast cells secrete fluid, creating a cavity, the blastocele, with the inner cell mass at one end. At this stage, the mammalian embryo is called a blastocyst. The cells of the inner cell mass eventually form two layers, the epiblast and the hypoblast. The epiblast is thought to contain the cells that will generate the actual embryo. Critical to the early development of all vertebrate embryos is the process of gastrulation, which occurs between days 13 and 19. Gastrulation results from the movement of cells toward the midline and then forward along the midline, forming a groove called the primitive streak. The end result is the production of the three primitive cell layers, the endoderm, the mesoderm, and the ectoderm. The nervous system develops from the outer layer, called the ectoderm. As this cell layer thickens, it becomes a distinct epithelium called the neural plate. Uneven rates of cell division form a groove called the neural groove that will become the midline of the embryo. Ridges of ectoderm continue to bulge on both sides of the midline. The tops of the neural ridges eventually come together, forming the neural tube. The interior of the neural tube becomes the fluid-filled cerebral ventricles of the brain, the central canal of the spinal cord, and the passages that connect them. By about 24 days, the three major subdivisions of the brain are discernible. These include the prosencephalon or forebrain, including the telencephalon and diencephalon, the mesencephalon or midbrain, and the rhombencephalon or hindbrain. The progenitor cells of the neural tube are known as neural precursor cells or neural stem cells. This represents the first stage of neural development, neurogenesis, when undifferentiated cells undergo mitotic divisions to produce either new stem cells or neuroblasts that will eventually differentiate into neurons. The cells undergo a stereotyped pattern of cell movements as they progress through the mitotic cycle. Eventually, the dividing precursor cells form a closely packed layer of cells called the ventricular zone. Eventually, some cells leave the ventricular zone, forming a second layer of cells called the marginal zone. Later, an intermediate layer develops as the wall thickens. Cells in the intermediate zone will then begin to differentiate into both neurons and glia. As the number of cells increases during the period of neurogenesis, the newly formed cells must move ever greater distances. The massive movement of nerve cells to establish distinctive cell populations represents the second stage of neural development, cell migration. Cells do not move in an aimless, haphazard manner. Cells in the developing cortex move along the surface of a particular type of glial cell called radioglia. Like spokes of a wheel, these radioglial cells extend from the inner to the outer surfaces of the emerging nervous system. The radioglial cells act as guide wires, and the newly formed cells creep along their processes. Once cells reach their appropriate destination, the third stage of neural development begins, differentiation. At this point, the cells begin to use or express particular genes. This allows the cell to acquire the distinctive appearance and functions of neurons characteristic of that particular region. As the process of differentiation proceeds, the fourth stage of neural development begins, process outgrowth. The biggest change in brain cells early in life is the extensive growth of axons and dendrites and the proliferation of synapses. The neural environment also greatly influences nerve cell differentiation. In vertebrates, young neural cells seem to have the capacity to become many varieties of neurons, and the particular type of neuron that a cell becomes depends on where it happens to be and what its neighboring cells are.
All right, that's it. Have a great day.